Hello everybody, my name is Enrico Massignani and the poster I'm presenting is titled MoDPA, Investigating Diseases Through Modification-Dependent Protein Associations. Proteins are the final product of gene expression and perform all sorts of functions in the cell. However, they do not work in isolation. Instead, their activity is tightly regulated through functional interactions with other proteins and also through post-translational modifications, or PTMs for short. These two mechanisms are very well known, but their interplay is still poorly understood. So to fill this gap of knowledge, I am applying network biology techniques not to a network of interacting proteins, but rather to a network of co-occurring PTMs. So first of all, how did I generate the network of PTMs that I'm working with? So one of the ongoing projects in the Compomics lab, where I'm doing my postdoc, is the reanalysis of all mass spectrometry data in PRIDE. So we downloaded about 1 billion spectra from PRIDE and reprocessed them with an open PTM search tool to identify PTMs in an unbiased way. For each detected modified residue, I calculated the fraction of proteins carrying that modified residue in each experiment, and I used this data to train a variational autoencoder. A variational autoencoder is a deep learning model that learns how to compress multidimensional data into a low-dimensional latent space, and then decompress this latent space to recreate the original input data. The idea is that the latent space will contain all the essential features of the original data, free of noise and possible artifacts. So to obtain the final PTM network, I calculated Pearson correlations between the different PTMs inside the latent space. So in this network that you can see here, each node represents a specific modification on a specific residue of a protein, and the edges connecting the PTMs are weighted based on their Pearson correlation coefficient. So during the preliminary analysis of the network, I identified several clusters of significantly co-occurring PTMs, and the cluster that I'm showing here is made of PTMs that occur on proteins involved in different aspects of the immune response. For example, phosphorylated proteins appear to regulate viral replication, whereas methylated proteins are involved in apoptosis and cell movement. And finally, oxidated proteins are the ones more directly involved in the immune response, for example, through the production of reactive oxygen species, which have an antimicrobial activity, but also end up modifying the protein themselves. So in the near future, one possible application of this MoDPA method would be to the study of chaperone interactions. So chaperones are proteins that can bind misfolded and damaged proteins to repair them, but PTMs can disrupt these interactions, leading to the accumulation of protein damage and eventually to degenerative diseases like, for example, neurodegeneration. So thank you very much for your attention. If you are interested in this project, or if you have any questions or suggestions, please do not hesitate to contact me. Thank you again.